When we talked about the cell cycle, within interphase we talked about the S phase, which is where DNA copies itself in preparation for the cell to divide so that each daughter cell has a copy of the DNA to run itself. We're going to get into exactly how that happens, the nuts and bolts of all that today. The base pairing that we talked about, the straight line letters, the A matching up with T, and the curvy letters, the C matching up with G, those base pairing rules that were discovered by Chargaff explained how DNA could be copied because of that unique base pairing that happens. Each strand of the double helix has all the information needed to reconstruct the other half because of base pairing. I'll show you how that works. The word that we use to describe the strands, because each strand can be used to make the other strand, is complementary. That's the word that we use to describe the two strands of DNA. The process by which DNA is copied is replication. And again, just to reiterate, this process happens to make sure that the daughter cells that come from cell division each have a complete set of chromosomes. During the process of replication, the DNA molecule separates into two strands and then uses the base pairing rules to form two new complementary strands. We call this original strand of DNA a template or a model for the new strand. We'll get into this diagram in the coming slides. So in the first part of replication, the first step is that those two strands of the original double helix separate or unzip. And that allows two replication forks to form, like a fork in the road. This is where the DNA is unzipped in the middle, and we have replication happening at both ends. This is actually called a replication bubble. New bases are added following the rules of base pairing, and the enzyme that adds those new bases is called DNA polymerase. Remember that nucleic acids are only one example of the different polymers we've talked about. So a polymerase is an enzyme that creates a polymer. In this case, it's the polymerase that creates DNA polymer. How this works is if the base on the original strand is adenine, then thymine is added across from it. And then again, guanine is paired to cytosine. So the base pairing rules are followed. The end result is two DNA molecules identical to each other and to the original molecule. Each DNA molecule at the end of replication has one original strand and one new strand. This is called semi-conservative replication. If you conserve something, you keep it the same. You keep it in its original state. So semi refers to half of that new strand is original, and half of it's conserved, and half of it is new. There are 3 different enzymes that are used in DNA replication. The first enzyme is a helicase. The helicase unzips the molecule of DNA, gets in there and breaks the hydrogen bonds between the base pairs. Each strand then serves as a template for the attachment of complementary bases. And remember, I said that DNA polymerase attaches the complementary bases. So that's the second enzyme, DNA polymerase. The other thing that DNA polymerase does is it proofreads. Quality assurance. Proofreads each new DNA strand, making sure that each molecule is a perfect copy of the original. There's another enzyme involved called ligase that links everything all together. Here, let me go back. Sorry. Ligase, once the DNA polymerase has added all the bases in, ligase links the strands all together, seals it all up. The tips of chromosomes are known as telomeres. The ends of these DNA molecules are really difficult to copy. DNA might get broken off each time a chromosome is replicated. An enzyme called telomerase adds short DNA repeats to the telomeres, lengthening the chromosome slightly and ensuring that when something gets broken off at the end, it's just junk DNA. It's not from a gene. We call this, like I said, junk DNA. Junk DNA is at the ends of the chromosomes and also in between genes. We'll come back to junk DNA later. Replication in prokaryotes is a little different because there's a single circular DNA molecule. It's way simpler, contains nearly all the cell's genetic information. Eukaryotic cells can have up to 1,000 times more DNA than prokaryotes. And if you remember, DNA of eukaryotic cells is found in the nucleus. In prokaryotes, replication doesn't start until regulatory proteins bind to a single starting point. It proceeds in two directions until the entire single chromosome is copied. So we have a circular chromosome in prokaryotes. We'll have two replication forks, and then the circular DNA is copied. The two chromosomes produced are attached to different points inside the cell membrane, and they're separated when the cell splits to form two new cells. Eukaryote DNA is more complex. There's much more going on. The chromosomes themselves are bigger, and there's more than one chromosome. So it's more complicated. In eukaryotes, replication can start at dozens or hundreds of places along the chromosome. So there can be multiple replication bubbles, like I called them, with two replication forks at each bubble. And they proceed in both directions until each chromosome is completely copied. The two copies of DNA produced by replication in eukaryotes remain closely associated until the cell enters prophase. At that point, the chromosomes condense, the two chromatids become clearly visible. They're actually connected together at the centromere, if you remember, and they separate from each other in anaphase of mitosis, producing two cells, each with the complete set of genes coded in the DNA. So in prokaryotes, the DNA gets copied, those two chromosomes get separated from each other right away. Eukaryotes, the sister chromatids that are the result of DNA replication, are actually tied together at the centromere, and then they separate at anaphase.